Good afternoon, everyone. In this lecture, we're going to learn about monocot and dicot embryo. So, let's start with the video. As you know, the process of the fertilization, wherein male gamete fuses with the egg cell to produce a diploid zygote. So, this is a zygote. It undergoes a horizontal division or transverse division to form two cells. One is a terminal cell, and the other one is the basal cell. Terminal cell will give rise to the embryo, whereas the basal cell it will produce suspensor. Okay, as you can see here, the basal cell, the terminal cell undergoes transverse division as well as longitudinal divisions to form globular shaped embryo, and this upper portion is, so this one forms the radical, and this one forms the plumule. Okay, this lower portion forms the plumule, and this upper portion forms the radical in this case. Okay, this is a globular shaped embryo, so it keeps on dividing to produce this kind of embryo. Okay, hypo means below the cotyledons regions. Okay, so if it is present below the cotyledons, then it is called as hypocotyle. If it is present above the cotyledons, then it is called as a epicotyle. Okay. So what is the function of suspensor here? The main function of the suspensor is it provides nutrients, specifically amino acids, to the developing embryo, and it also helps in positioning the embryo. It pushes the embryo towards the endosperm so that it can derive uh, nutrition from the endosperm. Okay, and also it helps in anchorage. That's all. So these are the three important functions that you have to remember. Then this is the structure of the monocot embryo. Okay, so you can see only one. This is a cotyledon, so we can find only one cotyledon in case of monocot, and that is called as a scutellum. This is a terminology that you should keep in mind, and this the protective this is plumule, which gives rise to the shoot system. Okay, and the protective layer which is covering the shoot apex, it is called as coleoptile. This portion is called as epicotyle. Okay, because if you divide the embryonal axis like this whole portion and the upper portion is called as the epicotyle, means it is present above the cotyledon, and it is this one is called as a hypocotyle. Okay. We can see some remnants of cotyledon in case of monocots, and that will constitute this layer that is called as epiblast. Okay, this kind of embryo is specifically found in grasses. The main function of this epiblast includes providing transferring nutrients as well as protecting the developing embryo. And it is also called as a rudimentary or vestigial secondary cotyledon. It is immature, reduced, that is specifically found in the monocots members. Okay, this is a root cap. You can see the root cap in case of monocots, and it will cover the radicle that develops into a, or it will produce a root system. The one which covers the radical, that protective sheath, is called as a polyorhiza. Okay, this is specifically found in case of monocots. This you should remember. Polyorhiza is not present in case of dicots. Let's learn about a dicot embryo. This is a zygote. Here also you can see the horizontal or the transfer division to form, wherein uh, wherein it produces two cells. One is the embryo cell and another one is a suspensor cell. Here also the uh, this one is a terminal cell which gives rise to the embryo. Okay, and the basal cell gives rise to the suspensor cell. The functions of the suspensor cell we have discussed in the monocot embryo while explaining monocot embryo. So that it mainly helps in transferring the nutrients, and it provides a structural support, and it helps in anchorage. Okay, so this keeps on dividing, the basal cell keeps on dividing to produce so many cells. Okay, it undergoes a transverse division, and the terminal, this tip cell is called as a posterior cell. So it is the main cell which is involved in transferring the nutrients, okay, to the developing embryo. So it provides the nutrients to this embryo. This one, posterior cell, is involved in the transferring of the nutrients to the developing embryo. Um, this terminal cells undergo first horizontal division to form two cells, and then it undergoes a longitudinal division to form four cells. Okay, so two tiers of cell or layers. So to form the stage, the 8 cell stage or octant stage, this will develop into a globular embryo. So this the terminal cell of this suspensor cell is called as a hypophysis. Here it is not mentioned, but you can once again. So this terminal cell of the suspensor is called as hypophysis, and this upper portion is of the or the top portion of this embryo forms the hypocotyl region, which encloses root cap, root tip. Okay, whereas another thing you can see in case of diploid embryo is it has the this globular embryo has a outermost layer called as the dermatogen, and the cortex portion forms the ground meristem. Okay, and this procambium forms uh, vascular bundles. Okay, apical meristem undergoes some modifications, and it is also called as dermatogen theory. It was proposed by Hanstein in the year 1870. So it has a outermost layer called as epidermis, and that is called as dermatogen, and the cortex portion is uh, called as ground meristem, or also called as peribrum, whereas the innermost portion is called as a procambium, which gives rise to the vascular bundle, and it is called as a plerome. So these three are distinct meristematic layers that you should remember. And this globular shaped embryo, again, it undergoes division to form a heart shaped embryo. So this heart shaped embryo, it undergoes further divisions, or it elongates to forms the two cotyledon, and this is a radicle, and these cells are called as a suspensor, and this is the haustoria. And one more thing here you should remember is this one is the plumule, and this is epicotyle. So it, it is this is epicotyle, and it is present above the cotyledons. So these are the two cotyledons that you can see in case of the dicotyledon, and this is hypocotyle, and this is radical, which gives rise to the root system, and this is a root cap. So that's it, friends. You can try to solve some of the multiple choice questions. I've listed 10 questions for you people. So please go through one by one. Let's try to solve this question. Which embryonic region elongates during germination in monopods? So it is a mesopotal region. What it does is it pushes the plumule towards upwards in case of the monopod so that it can germinate into a new seedling. Okay. Even a suspensor function is also helps in is also to push embryo towards at this point to derive nutrition from it. Okay. So that's it, friends. For this lecture, I hope you have understood. If you have any doubts, please let me know in the comment box section. Please don't forget to like the video. So if you have understood the video, then please hit the like button and please subscribe to my channel. We'll meet you in the next lecture. Goodbye, take care, bye bye.